I'm Andrew Singer, and welcome to the lecture on the drivers of AMR in the environment. The learning objectives are three: to recognize the diverse sources of pollution in the environment that can select for and maintain antimicrobial resistance, to recognize the diversity of chemicals that are known to drive the selection for and maintenance of antimicrobial resistance, and understanding how to evaluate the risks posed by antimicrobial resistance driving chemicals in the environment. There's some selected readings with the DOIs present for your ease. And now jumping into why even talk about environmental AMR. So we all exist within the environment where AMR microbes and AMR driving chemicals are discharged. And the environment is where AMR evolves, in addition to within us. And the risks from that evolution in the environment transmitting back into humans and animals is why we focus on the environment. So the major chemicals that most people would be thinking of when you talk about AMR driving chemicals are the antimicrobials. So those are antibacterials, antivirals, antifungals, antiprotozoals, and the antihelminths. These chemicals are used in the environment in many different ways. So agriculture, aquaculture, husbandry, animal husbandry. We consume all of these in our homes. A nice sort of subset of these chemicals are used in hospitals. And then, of course, the production of these chemicals and use of them in industry. Each one of them has a different pathway to get into the environment, but they all eventually do get into the environment. And they get into the environment via runoff, via effluent, via direct discharge. So aquaculture would be a direct discharge straight into the environment. And each one of these has a pathway that could take a varying amount of time, and in that time you could have the loss of the chemical. But also in that long period of time you can have selection for resistance in the sort of the journey of getting it from the source to the sink. So diving into some of the major sources of antibiotics or antimicrobials in the environment is, of course, the manufacturers. And in this particular case, I'm talking about antibiotic manufacturing. There has been a fair bit of attention given to this quite early on, because it's pretty obvious that antibiotics have to be produced somewhere, and that the manufacture of these creates a risk to exposure in the environment. And that risk had been previously unmeasured, but at least the risk has been characterized by a number of people going back into the 1990s. But really it didn't hit the mainstream academic and also policy world until maybe the mid-2000s, where some really horrible case studies were demonstrated, where downstream of pharmaceutical manufacturing facilities in India, ciprofloxacin was at levels that were as high as therapeutic levels of this chemical in human bloodstream, so about 31 milligrams per liter. And similar case studies were revealed all over the world. Most of these in modern times are being produced in countries where the regulations are not very rigorous. And so India and China are the primary locations where a lot of this kind of evidence is being generated. So the environment is being receiving all of these chemicals as a result of the manufacturing. The manufacturers create these in vats. They have to clean out the vats after each batch, and they can sometimes go through wastewater treatment plants, which have varying degrees of efficacy. By the look of much of the data, not very effective, and then it goes into the environment. And so you're basically then selecting for resistant organisms in the environment. There are calls for this to change. And so green manufacturing is one of those sort of buzzwords that captures this interest. And the interest then could be translated into making sure that the disposal of any residues into the environment are mitigated through effective wastewater treatment, or even better, no discharge to the environment. So Some of these solutions can be zero discharge, which means that they can desiccate any of the products they have. They could then burn it. Not very impressed, sorry about the font on that, but effectively what that is saying is that there are, on a national level, surveillance programs or antibiotic stewardship programs, which are quantifying the amount of antibiotics being used in animals and in humans. In the UK, the amount of antibiotics used in humans is roughly twice the amount that is being used in animals. That's not true for the rest of the world. The rest of the world tends to use more antibiotics in animals. The country's globally antimicrobial usage for food-producing animals is estimated at roughly 100,000 tons. So it's a massive amount of antibiotics. What's important to know is that only a fraction of the antibiotics consumed by any one animal is broken down in their body. So all of that 100,000 tons is finding its way into the manure and the urine, and that is effectively an environmental exposure. That's the pathway that it gets into the environment. So the next topic that I'll cover is black swan events. No pun intended that I'm talking about swine flu, which one of the natural hosts for influenza is in fact swans. So back in the pandemic of 2009, or in fact before the pandemic, we were aware of the fact that Tamiflu was going to be used in quite significant quantities before the pandemic had occurred. This was a new drug that hadn't been used before, and so the stockpiles of this drug were sufficient to treat, depending on the country, 25 to 50% of the population. So ahead of the pandemic, we did some studies to try to understand what's the implications of using so much of one drug that has never been used before all at once. So you're talking about several weeks worth of drug use, and that is massive. It turns out that depending on the size of the pandemic, you would get a different result. So in a pandemic of an R0 of 2.3, we predicted that the wastewater treatment plants probably wouldn't be able to cope with this load of antivirals. The reason that the antivirals would have an impact on the bacteria, which is the main function or operator within a wastewater treatment plant, is that the antiviral has actually been demonstrated to impact, and in a negative way, biofilm formation. And biofilms are pretty much the working horse, or the mechanism by which bacteria within a sewage treatment plant can actually do their job. So as soon as you reduce the efficacy of biofilm formation, you reduce the ability to treat sewage. And so we raised the issue that sewage treatment would be compromised during a pandemic if it was quite a severe one and everyone took those drugs. Turns out we also would use a lot more antibiotics because of secondary bacterial infections from influenza. And so this problem of risking our wastewater treatment plants is even further exacerbated by the antibiotic increase. Now, the downstream risks from that, other than having lots of pollution going into your rivers, is that the antivirals don't break down. Tamiflu is very persistent, and so it would go into the river as the active antiviral. And because swans and other wildfowl are the natural host of influenza, the exposure of the natural host with the infection and a drug that would select for resistance, all in the same place, predictably in every country, would really quite risk driving antiviral resistance in that wildfowl, which naturally is, because it's the natural host, is then a direct risk to us, which means that should we have a second or third wave of pandemic influenza, the likelihood of the drug working as well as it did the first time, should it have worked really well in the first time, would have been considerably reduced. So this actually did pan out. We did find resistance. Whether or not it came from environmental exposure or human use is unclear because it's quite difficult to know that. But there laboratory studies with wildfowl or ducks did demonstrate that selection can occur in the wildfowl from exposures of antivirals at environmental concentrations that we predicted. So that's the antivirals. the antimicrobial selection, the selection for resistance in the environment can occur in several different ways. You can have the spreading of the chemical, as I said with the Tamiflu, is the chemical exposure in the environment, but you can also have the spreading of the genes and or the bacteria that contain the genes. And that can happen a multitude of ways. One of them is through our pollution exposing wildlife to AMR, some sort of resistant organisms. And this occurs quite regularly. Where you look, you find that wild birds that cohabit our polluted environment will be carrying clinically important resistance genes. And so multi-drug resistance is regularly found in wild birds, and there'd be no reason why they should be carrying that other than from exposure to our pollution. Now, there is a bit of a question mark as to how much of it comes from us to them, and then how much of what they have comes back to us. And I suppose that's going to be different for everyone, and it probably will differ based on the birds. But it's not just birds. There's actually a lot of other wildlife where people have dived in. So during the pandemic, coronavirus was found in deer and almost every mammal, cats and dogs. So there's a wildlife segment, which is the recipient of our pollution, as well as potential source of AMR from which might have evolved in the environment. Another one is through the air, which is quite understudied. We know that the environment is obviously receiving our pollution, but where is the pollution coming from with regard to the resistant organisms that pollute our air? We don't actually know that. We don't know if there's selection going on in the environment, so in the particles that are floating around, the PM 2.5s potentially. There are surfaces that bacteria can lay on and create a biofilm or just cohabit. And when you sample these, you find it loaded with resistant organisms, not necessarily pathogens, but they're just resistance genes. And when you inhale these, they can be effectively assimilated into the microbes in your lung, in your throat, in your nose, in your gut. And this is something that really we're only just recognizing is a thing. And the extent to which it does all of these things, what impact it has on humans, is to be determined. Another big area, which is really quite a hot topic now, is the antifungals, because we've been focused on antimicrobial or antibacterials really since the beginning of our interest in fighting antimicrobial resistance. But we're realizing that antifungals are really quite important. And the antifungals are used in virtually every crop that we grow, especially in the UK. So there's some crops receive multiple sprays of antifungals. And what these antifungals are trying to do is they're trying to basically keep your crop healthy. But the downside of having done that is that there is a residue of antifungals on all of this organic matter, which may end off going to a composting facility. And this composting facility raises the temperature through its natural actions of composting to temperatures that are consistent with the perfect growing conditions for an Aspergillus fumigatus, which is a fungus. And this fungus, which would normally just be decaying plant matter, and that's essentially what it is doing in a compost bin, but it's also having to decay this organic matter in a sea of antifungals. And so the only way it's going to be able to achieve this degradation is to be resistant to the amount of antifungals that it's being exposed to. And so it develops resistance while in the compost bin. And then because Aspergillus disseminates itself through spores, it then releases spores. And what we didn't know until a recent study, which is shown here, that there are going to be hotspots across the UK and across the world wherever you have these composting sites, and these composting sites receive waste material that has actually had azoles, which is one of the antifungals that can drive resistance. Now, why is this important is that azole resistance has been showing up in the clinic. And you go back into the 1990s, there was virtually no such thing as an azole-resistant aspergillus. But now you're finding it in 30 or 40 or 50% of the azole-resistant, or in the aspergillus infections, aspergillosis. Depending on where you are, it could be even a lot worse than that. So you have virtually no solutions for treating some of these aspergillus infections, and it's because of our use of antifungals in the agriculture sector. So this is something that is now being studied, or has been studied, but is being studied even more now in the United States, where they examined azole-resistant aspergillus where they found them in 7 different states of the 8 that they looked for, and 27% of the isolates that they found were pan-resistant, which means that there was no way to treat them if they were an infection. So one of the probably big sources of maybe traditional antimicrobial resistance in the environment is wastewater. And wastewater is, the composition of wastewater reflects the people who are upstream of it, the drug habits in the sense that what infections they have, what are they treating themselves with, whether there's a hospital there. Hospitals use antibiotics that we wouldn't necessarily use at home because they needed to be injected into your bloodstream. And there's a lot of other drugs being used in a hospital that you wouldn't be using it in your home. You obviously have industry, and so all of this is being mixed together in effectively a bioreactor. They were all just with the microbes and the chemicals all interacting, and there's almost no way to make this a better place for AMR to be selected for, to mix, to recombine, and you get virtually every resistance gene that you want will be found in wastewater. Now, so there's been some work been done on that in the UK, where we looked at 10 different sewage works and the amount of antimicrobial resistance that exists at any one point in a sewage treatment plant, and to what extent do these different stages of treatment affect the fate of a resistance gene. And by and large, treatment is a good thing. So that's probably the one confident thing you could say about wastewater treatment with regard to AMR. You have less AMR coming out of a sewage works once it's been treated versus the amount that came in. So that's what one of these global multinational studies has shown, where they looked at cefotaxime-resistant coliforms, and they basically showed that less of those come out than went in. But the problem is that there's still quite a lot that comes out. So about 1 million of these resistant coliforms are coming out in every liter of final sewage. And that's not what you'd call treated to safe levels, I suppose. If you have receptors downstream, like a bathing water, 10 to the 6 bacteria per liter of resistant coliforms is a lot. The next study that I'm highlighting here is when you look at wastewater and you see the suite of resistance that is found within it, you might want to ask the question, what is the relationship between the resistance that you find in a wastewater and the resistance that you find in the population? And so there has been a study, the systematic review, to look at that, and they find that actually the relationship is quite good. There will be differences because, as I say, there's a lot going on in wastewater. So there's a lot of genes mixing. There may be some selection going on. There almost certainly is some selection going on. And all of these will change the suite of resistance genes and the proportion of them from the point where it came out of you and gets collected by a scientist like me. Okay, changing gears a bit. We may be familiar with the Sustainable Development Goals. They've been around for quite some number of years now, and they're coming to an end in 2030. And one of those goals was having the proportion of untreated wastewater going into the environment, which was anything that would decrease the amount of wastewater going to the environment. Now, you could be aware of the fact that there's a lot of AMR in wastewater, and so by reducing it, you're doing a good thing. And this is the state of play back in 2022, the way that the WHO is presenting the world, the proportion of wastewater flow safely treated. And so you can see this is just a handful of countries on the right-hand side and the whole globe on the left-hand side. And so if you're red, you're not doing very well. So it's projecting that you must have somewhere between 0 and 50% of your wastewater being treated properly. And if you're green, then you're treating 75% to 100% of your wastewater properly. So you can look at something like that and be sort of reassured that you live in a country that's green. But what I'll show you is that if you dive into these numbers, you then start to realize that actually maybe these numbers aren't as valuable as we thought they were. Or maybe they're saying something different from what we thought they were saying. And so I'm going to dive into just one of the ones that was ranked at the top. So Gibraltar was one of the countries that, well, debatable whether it's a country, but Gibraltar was at the top of this list, along with Kuwait, Singapore, and Andorra, where they say that 100% of their wastewater is safely treated. And if you dive into that and you look at the literature and the reports on what are they treating, how are they treating it, you realize only last year, there's an article in the newspaper saying that EcoWaters here says plans are well underway to deliver Gibraltar's first wastewater treatment plant, which could service the rock for the next 50 years. The absence of any such facility in Gibraltar has drawn both local and international criticism over many years, with raw sewage pumped out directly into the sea. So you say, well, if 2023 reports that the sewage treatment plant hadn't actually been up and running yet, but in 2022, they're reporting that all of their sewage was treated safely, something isn't matching. And so what it then makes you question is, how good are these numbers that are being reported at national levels? And I would say they're probably quite bad, because just in the UK, we know that a huge amount of sewage is being untreated and discharged into the environment. And that amount of sewage is percentages of a daily flow of sewage. So probably somewhere between 1 and 5% of all the sewage being produced nationally is going to be going through a pipe and exiting into a river being untreated. Several years ago, we did a machine learning paper that exposed the reality that sewage was being discharged into the environment in quantities that were being unreported. And if they're being unreported, that means that the WHO numbers that I just presented to you were lofty overestimates of the amount of safely treated sewage that was going into the environment. And so as this developed over the past few years, some of the water companies are now reporting in real time how much sewage is actually going into the environment. And so this is just a snapshot of what was going on one of the days in the Thames water region. And every red dot is an indication that untreated sewage was going into the receiving river. In general, in 2023, England and Wales saw 464,000 individual spill events, which equates to about 3.6 million hours of spills from storm overflows. If you were to add all those spills up, it equates to about 410 years worth of sewage continuously discharged into just England and Wales. So there's a big problem that is brewing underneath these national statistics. Now, it's not something that we're unaware of anymore, but I think what other countries will need to do is ask the same question: how much of their assumptions are correct? So here's a video just to sort of reinforce the point that this is considered treated sewage by the regulator, the environmental regulator. This is a video taken by Ashley Smith at Windrush Against Sewage Pollution, who have been bringing to light some of the evidence that treated sewage does not look the way it should look. And so this is what's coming out of a treated sewage well, this is treated sewage as far as the regulator and the water company is concerned, but it clearly should not be looking like that. And there may be reasons why it looks like that, but they're not going to be good reasons, because treated sewage should never look like that. It won't be drinkable, but it should be something that poses the environment very low risk. And that's a journey that we need to go on. So as far as risk is concerned, we have very few studies that quantify risk in using real data as opposed to completely modeled data. And so one of the few studies that does this is called the beach bum survey. And so they look at effectively swabs of the bums of beach surfers, and they look at a control group that doesn't go in the water. And they found that 11 of the 97 bathing waters themselves were sampled concurrently with the bums, and found 11 of the 97 found CTXM bearing E. coli, and that the surfers themselves, who exposed themselves quite regularly to this kind of water, were 4 times greater than the control group to carry those organisms, and 3 times greater to carry cephataxime-resistant E. coli as compared to the non-bathers. So this is quite a rare study. We need a lot more of them, but it shows that exposure, or chronic exposure, but volunteered exposure, because it's a recreational activity, is a risky event for acquiring a resistant organism that could actually be important one day down the line for that person who's carrying them, because if they get an infection, they already have the gene that might be preventing that drug from curing an illness. And that can also be transmitted to another person, maybe in their family. So up until now, I've been talking to you about the amount of antimicrobials in the environment and the impact that they have, because that's kind of the first line of we need to get that sorted. But there's a second line of we need to get it sorted, which are basically all the other chemicals that we use. When you look, what you tend to do is you find. And so if you use metals, biocides, herbicides, fungicides, and pesticides, and really non-antibiotic pharmaceuticals, any one of those, if you expose them to either a pure culture, maybe an E. coli, or a community of bacteria, maybe sewage, and you have metrics of what change looks like, and everyone tends to have a different metric of what change looks like. It could be the prevalence of a gene, it could be respiration, it could be all sorts of different endpoints. But what you find is that when you use these chemicals, you get an effect concentration, and eventually, because basically too much of anything will start to have a negative impact on a microbe. But when you get effect concentrations at environmentally relevant concentrations, you then start to ask really important questions of, well, where in the world do we get these concentrations? And are they co-selecting for resistance, antibiotic resistance or antimicrobial resistance? Because basically a chemical will be toxic to a bacteria. The bacteria has to come up with a solution to that. And if the solution is the same solution that it has for an antibiotic, you're co-selecting for an antibiotic resistance. And these are the terms that we use, which is co-resistance and cross-resistance. Cross-resistance is when the same solution can actually prevent you from having the toxic effects of multiple chemicals. And co-resistance is when the genes are linked on the plasmid, usually a plasmid. And so by maintaining a gene, because of one chemical being present, you're actually also maintaining another gene. So there's also another set of terminology that we use in environmental microbiology, and it's called the predicted no effect concentration for resistance. And the reason it's important is because PNEC, predicting no effect concentration for resistance, is a term that you could imagine a regulator would use to determine what safe looks like. And so what safe looks like with regard to resistance is what is the concentration in the environment that doesn't pose a risk but isn't overly protective, meaning that you're getting as close to risky as possible without getting there. And so to derive that kind of number, you need to do either lots of experiments or you use models. And so here I'm explaining how you would get to that using a model. So there's something called UCAST. UCAST is basically a database of the amount of resistance or tolerance of any one microbe to any one antibiotic at any one concentration. And so you get these distributions of effect. And so here's sort of on the top left, you have amoxicillin and then cefotaxime and all the other antibiotics. And each one of those dots is a different microbe. And so you have a species sensitivity distribution. And then you have to say, well, there's a concentration of amoxicillin, for example, that is sufficiently low that 99% of the bacteria that we're aware of would be unharmed if exposed to it. But the thing is that because UCAST is actually describing minimum inhibitory concentrations, which is the concentration that actually kills them, you're actually still killing about 1% of the bacteria because you've protected 99% from death, but they're still having negative effects at concentrations close to that. And so what you want to do is back off from that. And so a safety factor backs off from that minimum inhibitory concentration and moves it to something that you call, it's more protective because you assume that although you may have 15 to 30 different microbes that make up this species sensitivity distribution, that's only a tiny, tiny fraction of all the microbes that exist on Earth. And so you want a bit more confidence that you're protecting all the ones that you're not measuring, but you also want to recognize the fact that the value that you're using here is actually completely inhibitory, and you're just looking for selective enough so that the resistance gene is favored over a non-resistant organism. And so once you do these calculations, you're left with concentrations that are potentially regulatory or of a regulatory value. And so there are data sets that exist that explain what these numbers are for every antibiotic. And using a model obviously means that these numbers are untested, so we don't know if it actually holds up. And so that's what people do is they do environmental exposure studies. They do on single microbes and combinations of microbes, on mixed communities and sewage. And by and large, you get roughly the same kind of answer depending on, regardless of how you do the study. They tend to gravitate to a similar number. And much of the time, those numbers are very similar to the modeled numbers, but occasionally you will get outliers for some of the antibiotics and where the model was much more protective than it needed to be. And so if you were regulating, you were probably, and it plays out like that, then you might be over-regulating for some antibiotics. And so we need to sort of combine lab-derived data to make sort of the ground truth and test whether the models are good representations of reality. So one of the things that you'd want to know is how bad is it? Is our world already chock-a-block full of antibiotics at concentrations that exceed these predicted no-effect concentrations of resistance? And so someone has already done this, and here's a study where they compiled not extensive, but enough evidence from different categories of the environment, so municipal wastewater, hospital wastewater, industrial surface water, drinking water. And so fortunately, virtually, in fact, zero of the drinking water exceeded these concentrations of resistance generation. But as you get more into the wastewater world, you start to see, so surface water is contaminated with wastewater, and so you get roughly 2 to 3% of all of the samples that were taken in the study or across the survey were in excess of the selection concentrations. And so this gets a lot worse in industrial wastewater and in hospitals, and municipal wastewater was somewhere in between. So what the main message of this is, the environment does have concentrations well in excess of the concentrations needed to select for resistant organisms. So what do you do with that kind of data? So one of the many things you can do with it is to see how it can inform your antibiotic stewardship. So if antibiotic stewardship was sufficiently constrained such that the waste that we produce didn't select for resistant organisms once the wastewater exited, well, once it got into wastewater, and then once it exited into the receiving river, then you have a really nice and tidy way of linking up antibiotic stewardship to environmental selection. So this was a study to try and do that, where we looked at the amount of antibiotics being used within the two classes, macrolides and quinolones. And so at day 0, which in this case was 2015-16 data, the amount of antibiotics falling into these two classes created an environment where selection was very likely to be occurring in 63 to 73% of the entire length of the Thames catchment, which is shown in the picture. All of the black dots were sewage works, and so we knew that the population's sewage goes into those points, and then those points are sewage works which discharge into rivers of varying size, and so the smaller the river, the less dilution is there, and the higher risk that would stem from that. So if you look at this in a sort of granular way, you'll find that rivers that are colored red are usually quite upstream, because the upstream rivers are the smallest rivers. And as you go further downstream, you have more water to dilute your pollution. So the question then is, how much stewardship needs to be enforced, or what does the stewardship level need to be at for us to get to a river where selection is not generally occurring? And that number is roughly 77 to 85% of 2015-16 prescribing needs to exist. So we need to cut out 77 to 85% of the prescribing that we're currently doing, given the kind of hydrology that we understand to exist today. Obviously, in a changing world where maybe rivers won't actually have as much water as they do today, that number would actually get even worse because there's less dilution. I set this model when we did the study at, we want to protect 90% of the rivers, because a notable thing about UK rivers, and probably it's going to be a global issue, is that some rivers only exist because they receive sewage. There wouldn't be any water if not for the sewage effluent. And so you can't really do anything about that other than to get the amount of antibiotics in the sewage to be below the predicted no effect concentration. And that's going to be a difficult thing, but it's not impossible. So I'm going to finish on a slide that's talking about the global factors controlling environmental AMR. The global factors include population increase. So obviously, as there are more people, they will be taking more drugs. They'll be eating more food that required more antifungals. The fact that any local selection that might be going on has now become an international problem, because we get on trains, we get on planes, we get on boats. And basically people move around. I think we're all pretty comfortable with this kind of phenomenon after the pandemic. The fact that there aren't any backup solutions, so if we lose an antibiotic, because a bit like the antifungals where there's pan resistance to the antifungals, if there's no antifungal to replace it, you're stuck. So there needs to be a pipeline of antibiotics and antifungals and all the others constantly coming up to replace what we lose. Now, obviously, we would love to have a world where we don't lose any, but that's not the world that we live in right now. The warming planet also means that there's some indications that a warming planet is not good for infections, that you actually get more antibiotic-resistant infections in hotter climates. It's unclear why, but it's a phenomenon that we are observing. The behavioral factors, we basically live in a world where we have already nested or rely on antibiotics or antimicrobials to deliver the kind of healthcare that we currently enjoy. At least some of us enjoy it, because obviously access is limited in most parts of the world, to be fair. But where it is accessible, our behavior effectively relies on the fact that we know that if we get a cut and it starts to get infected, we just go to the doctor and we get an antibiotic. So this is something that we know can happen. It wasn't always that way, which is worth saying. Food animals, before antibiotics, there were no antibiotics being used to produce our food. Now there are, and we rely on that. And so we can't just take it away without having huge effects on our provision of food, both animals and crops. So if we decided tomorrow that antifungals or the azole antifungals should not be used on crops, which sounds like a very sensible thing to say, what are the implications of that? Would we have 30% less food to eat, or maybe even more than that? And then who are we making hungry, or worse? So then that effectively feeds into the inequality factors, in that the low and middle income countries have poorer sanitation, and their infrastructure ensures that they are exposed to more pollutants than people who live in high-income areas or countries. Access to clean drinking water, it's been a given for a long time that we know that's important, but if your drinking water is polluted either with microbes or chemicals, you're at a greater risk of harboring AMR. And then, of course, the last one is the investment. That the clean water, drinking water, requires investment. It doesn't just happen, and it requires sustained investment. You can't just make a clean drinking water and not be there to make sure that the pipes are intact and they're not sucking in sewage, which in fact is what happens in many parts of the world, is that the pipes go in, you leave them for a fixed amount of time. Some countries obviously will have earthquakes and just subsidence and all sorts of things that go on underground. And the pipes crack, and then you get mixing of sewage and drinking water. And then if you don't maintain those pipes, you don't have drinking water that is safe. Sustainable pharmaceutical manufacturing, I think I've already explained how that's important. Livestock biosecurity is an investment in the sense that you could have free-range chickens, but when avian influenza is spreading across the world, the suggestion is to keep your chickens indoors and make sure that they have no exposure to the wildlife, because that's where they're going to get it from. So biosecurity will prevent infections, or good biosecurity will prevent infections, and if you prevent an infection, then you don't need an antibiotic. And when you prevent infections and the numbers of chickens are in the tens and hundreds of millions, that's quite a lot of antibiotics that you avoid using. And it's not just chickens. And the last one is sewage. We treat sewage as well as we currently do, but the problem is we don't necessarily know what that means. People aren't really asking until now, in some places, these tough questions of, are we treating our sewage good enough? What percentage of the sewage we thought we were treating, we're not treating? Where is it going? And what's the impact of that? Many parts of the world don't even have sewage collection, and some of the ideas might be to, well, let's bring it to them. Well, it's one thing to bring sewage infrastructure to a place that doesn't have it, which sounds like a very good thing. But if you don't actually keep it, keep investing in it, not only in the infrastructure to make it work, but also the skills. Someone needs to know how to run it. And sewage works are not just they run themselves, because they're often needing quite a lot of expertise in engineering to keep them going. And with that, I'll jump into the further reading. There's 4 papers. I tried to spread it out across topics, the antifungals, general antimicrobial resistance in the environment, some of the systematic reviews, and the aquaculture is an interesting one, which you can dive into if it interests you. So with that, I'll just say thank you very much, and And also thank you to EPSRC for funding the UKRI Digital Health Hub. And with that, I thank you very much for your time.